Hi everyone. Today I'm actually just going to go through a little bit of an introduction to microorganisms. This is just going to be a very broad introduction to the different groups of microorganisms that exist in the body, the different relationships that can exist, and also at the end, just a little bit of an introduction to Koch postulates. So firstly, there are 6 different types of microbes that exist. These are viruses. So think about your norovirus, HIV, HPV, protozoa. These are actually eukaryotic cells, unicellular eukaryotic bodies that, as we know, similar to humans, have a very similar structure. Then we have algae, fungi, which can be anything that, for example, causes thrush. I'm just going to draw a mushroom, or try to draw a mushroom here. We also have bacteria. These are what basically cause infections similar to viruses. And finally, we also have archaea. Archaea are special in which they are called extremophiles. They love extreme temperatures, in which they are called thermophiles. They can also live quite happily in highly salty environments, in which they're called halophiles. And they can also live even in areas of low oxygen, where they also can make methane, in which they're called methanogens. So, microbes. These are the 6 types. And microbes can also actually be present in the body. And we give a special name to these microbes. These microbes are actually called normal microbiota. And believe it or not, they're present on a lot of surfaces in the body. They're important in not only disease, but environment, in the industry and agriculture. And normal microbiota are a population of microorganisms that essentially infect the body but don't cause any disease. So there's no disease. And they're present on what we call external surfaces in the body. Now you may be wondering, what does that mean? External surfaces, quite surprisingly actually, doesn't only include your skin, or what we traditionally call external to the body, but also includes parts of the body, including the GI tract, the respiratory tract, and also even tracts in the urogenital organs, and many, many other places. And why are these places called external surfaces? Well, if you think about it, and if we take the GI tract as an example, it's essentially just a tube that passes from your mouth to your anus. And if you really do think about it conceptually, it's just open to the external environment, because you take in food from the external environment and essentially defecate into an external environment as well. So the GI tract is open as an external environment. And that's why there are microbiota present. We also have internal surfaces where there are no microbiota present. And these are also called sterile surfaces or axenic surfaces. And the main reason why microbiota can't survive there is that they're essentially closed off to the external environment. These may include alveoli or even seminal fluid. Let's focus back on external surfaces because actually, interestingly, there are some external surfaces that do not contain any microbiota. These include the stomach, for example, and also the bladder. So why do these particular places don't have any microbiota present? Well, the stomach, as we all know, is a highly acidic environment. So microbiota are usually not able to survive in this environment because of the extremely low pH. And a similar argument lies for the bladder. So going back to the actual concept of microbiota, there are two main types of microbiota that we find in the body. One, a resident microbiota, and the other are called transient microbiota. And as their names suggest, resident microbiota are present permanently or for an extended period of time in the body. These can include E. coli in the large intestine, for example. Transient microbiota, on the other hand, are microbiota that are not present or only present for a very short amount of time if they have a particular purpose, for example. These microbiota can include some streptococci, or even diphtheroids. So you may be wondering, so as I mentioned before, these bacteria don't cause any disease, but what's the benefit? So there can be many different benefits of these bacteria present in your body. Firstly, normal biota of the intestine, if we take E. coli for example, are quite important as they allow for the digestion of vitamin K. Because our body is unable to digest some vitamins, some polysaccharides, and even some small fatty acids, these bacteria come in handy, and in addition to other microbiota, they're very helpful in order to allow for the digestion of food and ensure that the body can attain as much nutrients or as many nutrients as possible. Furthermore, we come to the concept of mutual antagonism. Now, mutual antagonism is a quite important concept in the study of microbiota, because it's essentially one of the defense mechanisms that the body has. Let's start off by explaining it with an analogy. Say, for example, you're at the beach and you found a very, very good spot on the side of the beach. Obviously, because you've pitched a tent there, or your beach umbrella, you are able to have fun. But say, for example, someone else comes and tries to take a spot. Obviously, they won't be able to because you've already taken that spot. And essentially, they are not allowed to be anywhere within this space where you are present. And the same concept is present with mutual antagonism in the body. As if we take the example of E. coli again, as E. coli are already present in the colon, and say this is the colon here with the lumen, as E. coli are already present, they're essentially already taking up all of the nutrients, space, and other possibly beneficial aspects of being in the colon of a person, and don't allow for other harmful bacteria such as maybe salmonella or any other type of bacteria that may be harmful for the body from entering and therefore causing any damage. And this is what's also called competition between different microbiota, because essentially they're both fighting for the same place, but as there are already your own microbiota present here, they're essentially protecting you from any other any other bacteria from taking your place. So that takes me on to the different types of relationships that can occur in the body. So there are four types of relationships that can occur with microbiota. The first is called commensalism. And commensalism is quite an important process because it's what people would say most bacteria are present in the body. It's essentially when two organisms are in a relationship and one organism benefits from it, whilst the other does to some extent or does not benefit from it. That doesn't mean that they are affected in any way negatively. They just don't have any worth in that relationship. So benefit to small extent for both, or there's no benefit for one, but benefit for the other. And once again, I'll take the example of E. coli, because E. coli in the colon essentially is quite happy there because it's receiving all of the nutrients that pass from the food that passes through the colon, but at the same time, it doesn't really have any benefit to the body other than allowing for the digestion of vitamins. Another example of this is Staphylococcus epidermis, which is present on the skin. And this bacteria essentially is able to take nutrients for its own benefit, for it to survive, but it doesn't have that many benefits to the actual body. Of course, it does, as I mentioned earlier, allow for mutual antagonism to occur. However, other than that, there's no real benefit. The second type of relationship that I'd like to go through is mutualism. Now, mutualism is basically a positive relationship that occurs between both organisms. This is when both organisms benefit from the relationship. And this can be seen with the many bacteria that are present in the colon, in the large intestine, as many of these bacteria allow for the fermentation of sugars, the breakdown of vitamins, and many other processes that allow for their own survival, but also allow for the human survival and essentially the reception of nutrients. The next relationships that I'd like to go through is called parasitism. Now, this is a relationship that usually is negative for both organisms and can end in three different manners. Firstly, parasitism can end with the host's death. This essentially means that the parasite that has infected the host is able to kill the host by its own proliferation, and in the process kills itself, because essentially as the host is dead, the parasite is no longer able to survive. The second way in which the relationship can continue is when the parasite dies. This is when the host is able to fight off the parasite and therefore survive, or albeit there may be some damage to the host, and the parasite is essentially removed. And the final and most interesting way this relationship can continue is that the parasite, although it's present in the host, is able to allow for the host to survive. And you may be wondering, how is this a parasitic relationship? Well, essentially because the parasite is taking away nutrients from the host, but at the same time, as they are allowing the host to survive, they can be passed down to different generations. Because as we all know, microbiota can be passed down from the mother to the fetus during both the formation of the fetus through placenta, or it can also be after the baby's born through breast milk. The interesting thing in this relationship is that as the parasite allows for the host to survive and live, eventually the host and the parasite go through the process of co-evolution. So eventually, the parasitic relationship, in fact, turns into a commensal relationship, or commensalism. Because after many generations, the parasite, although it's taking away nutrients from the hosts, is actually allowing for the host to survive and therefore is able to live with the host with minimal benefit to the host, but at the same time, great benefit to the parasite. Now, these relationships can have many different effects, and in terms of microbiota, they can have both positive and negative effects. So firstly, I would like to go through the idea of opportunistic organisms. And this is, I guess you could say, a fifth type of relationship, but it could also result from parasitism, or I guess even, a fourth type of relationship, and it can result from parasitism to some extent. Opportunistic organisms are organisms that are already present in the body, so they're microbiota, normal microbiota, but because of a certain change in the person, maybe they become immunodepressed, so they may have HIV, AIDS, they may be undergoing chemotherapy, or may have different conditions that essentially degrade their immune system. So the microbiota, because there's essentially no immune system that is able to control them, become opportunistic and go from maybe being commensal or mutualistic to negative and therefore pathogenic. And this is when we have infections such as thrush, or maybe even if, for example, E. coli moving from the colon to, say, for example, the small intestine, causing diarrhea and vomiting. And that's essentially when the mutual or commensal relationship becomes negative. And there are many, many different, I guess, things that we can do to combat that. And they include giving antibiotics, whether they be narrow spectrum or broad spectrum, or even other mechanisms that may allow for the patient to be back to their normal balance of microbiota. So that really concludes the relationship and the introduction to different types of microbiota in the body. The last part that I just want to go through, which is a little unrelated, but relates to microbiota as a whole, is what we call Koch's postulates. And Koch's postulates are quite important because they're essentially the building blocks of how we're able to research different microbiota. And there are four main postulates. And these postulates define how different microbiota are to be studied and whether they should be essentially categorized as being pathogenic or not. So the first postulate is that the causative microorganism must be present in every single disease and absent from healthy organisms. So all of the diseased organisms or humans, diseased humans, must have the pathogenic microorganism or microbiota if it's opportunistic. And the healthy humans should not, which makes sense because if, for example, a person has a throat infection, they will have a certain type of bacteria present in their throat, whilst people who don't have a throat infection won't have it. So that makes sense. The second postulate states that suspected organisms from the diseased host must be isolated and grown in pure cultures. What that basically means is that in order to test the presence of a microbiota, we must be able to isolate them from the diseased person and grow them in a culture. And a culture is just I guess, even it could be a petri dish with at a certain temperature and certain chemical environment that allows for the bacteria, viruses, archaea, algae to grow and proliferate. The next postulate states that the cultured microorganism should cause the disease when introduced to a healthy host. So after we've grown them in a culture, when we introduce them to a healthy host, they should cause the disease. Okay? So just going through the postulates once again. First, we saw the microorganism in the diseased human, but they weren't present in the healthy humans. Then we took them away from the diseased humans and put them in the culture and allowed them to grow. Then we took these organisms that we grew in the culture and introduced them to a healthy human, and they should have caused the same disease that they did originally in the diseased humans. Okay? And finally, the same organism that we took from the healthy host should once again be isolated and grown once again in a culture. Okay, so to go through all of them once again, the microorganism should be taken away from a diseased human and not be present in healthy humans. It should be isolated from these diseased humans and grown in a culture. Then, this isolated organism must be taken to a healthy host, introduced to a healthy host, and cause for the same disease to occur. And finally, the organism must once again be taken from a healthy host, isolated, and grown in another culture. So this is what Koch postulates are, and they essentially are what define almost all illnesses and diseases in the world. So in conclusion, this is basically a very broad overview on microorganisms in the body. There are 6 types. They can be present only on external surfaces in the body. They can have 4 main relationships. They can have 4 main relationships, as well as there being resident and transient microbiota. They can help in protecting the body. They can, in certain special situations, cause negative effects, and they also can be tested to cause disease in 4 small steps.